Hello, my name is Phil Tsao, and I'm sitting with this year's 2025 Jeffrey Haig ATVB Basic Science and Clinical Prize Award winner, Dr. Mark Feinberg from the Brigham and Women's Hospital. Welcome and congratulations, Mark.
Thank you so much. Truly honored and humbled to be the recipient of this award and to be here to present tomorrow.
As you know, the award is named after Dr. Haig. Dr. Haig, who was Chief of Cell Biology at the NHLBI, and is given to an investigator in the prime of his or her career. And certainly, Mark, that fits you well. So you're going to speak to us tomorrow, and why don't you give us a little summary of what you're going to speak about?
Sure. My lab focuses on the role of non-coding RNAs to help us understand the pathobiology of atherosclerosis and its complications, including ischemic injury in the heart and in the limb, with a major focus now in diabetes, given the disproportionate higher risk that we see of cardiovascular complications in our patients with diabetes. We have two different types of non-coding RNAs that we study in the lab, microRNAs and non-coding RNAs. And we study these non-coding RNAs in two different areas, one in the macrovasculature in the context of diabetes-associated athero, and also in the microvasculature in the context of impaired angiogenesis in ischemic heart in the limb, with the idea that these non-coding RNAs may help to uncover the biology underlying some impaired repair mechanisms that we see in patients with diabetes. I'm going to highlight two examples of non-coding RNAs tomorrow. One will be a link RNA that we've termed Miracle for macrophage-enriched link RNA that regulates chemotaxis, inflammation, and athero. What's really interesting about this link RNA is that it increases with diabetes-associated athero. It controls macrophage chemotaxis into the blood vessel wall in an epigenetic manner. What's really interesting is that when we knock down the expression of this particular link RNA, there was no effect on insulin resistance or diabetes. There was no effect on obesity. There was no effect on the lipid profile. And actually at this point, remember some chatter in my lab, so we even bothered looking at atherosclerosis itself, which was the main goal of the project. And it turns out it was one of the most potent reductions of atherosclerosis we've ever seen in the lab, about an 85% reduction in athero in the aortic arch. So hence the name link RNA Miracle, because we didn't expect it to have such a profound effect in a very harsh environment. Second part of the talk, I'm going to provide an example of a microRNA and how this regulates the microvasculature in the context of diabetes and chronic limb-threatening ischemia. What's interesting about this microRNA that I'm going to highlight tomorrow is known as the cis antisense microRNA. It regulates a target on the opposite strand of DNA, and it targets a process we had not thought about before. It targets protein aggresomes or aggregates, which turn out to be higher in diabetic skeletal muscle endothelial cells. And when in cells in a dish, we showed that this microRNA, when you overexpress it, reduce the number of these aggresomes in diabetic endothelial cells, and at the same time it promotes angiogenesis. And what was interesting in vivo and in human subjects, this microRNA was reduced in ischemic limbs or in peripheral artery disease, hypothesizing that perhaps this might be a protective microRNA. So we went ahead and delivered this intramuscularly, and it reduced agrosome formation, it promoted angiogenesis, and it restored blood flow recovery in the diabetic limb model. So highlighting that these non-coding RNAs can really uncover new aspects of biology that we had not anticipated before.
Wow, fantastic. Well, we've long known that there's much in terms of the micro and macrovascular complications associated with diabetes. What have we learned in your work in terms of similarities as well as distinctions between those two worlds?
Yeah, it's a great question. We learn a lot, obviously, from our patients. When we see patients with diabetes and complications, both the macrovascular and the microvascular are not separate entities, that they both coexist with their impaired atherosclerosis, accelerated atherosclerosis in patients with diabetes, and at the same time they have impaired angiogenesis. And it's very humbling because it's a very harsh environment. I think we've learned quite a bit that for many years there have been clinical trials that have tried to introduce vascular growth factors to restore perfusion in the heart and the limb. And from a lot of data that our group and others have looked at, utilizing a lot of the non-coding RNAs as tools to uncover the microvasculature, we may have gotten it wrong. Some of the microvasculature we appreciate, permeability is not a good thing. We're trying to improve permeability, whether it's in the heart and the limb, and we have some other non-coding RNAs that we're actually working on. I'm not going to have time to talk tomorrow, that can restore permeability, and we hope that may be a new avenue of direction, and in part learned from our patients in many clinical trials over the years.
Well, the Hoeg Award is given for excellence in both basic as well as clinical science. You actually travel in both worlds as a physician scientist. Tell us a little bit about how important it is and the approach that you take in the basic science lab and how you translate that into clinical manifestations.
Yeah, so first and foremost, the questions that we ask are inspired by the patients that we see in front of us at the bedside, but also we recognize for almost all our projects, we've been focusing on evolutionary conserved non-coding RNAs. So those that are derived from human samples, whether it's in the plasma and the blood, where you can detect these non-coding RNAs in circulating microvesicles, or specimens from biopsies from patients, and then to reverse translate that back at the bench to understand its role mechanistically in cells, and then in vivo using RNA-based delivery approaches, and then validating these non-coding RNAs either as biomarkers or as mediators of therapeutics using whale phenotype samples. And then the ultimate goal is to bring it back to patients. We're currently working on trying to do that, but that's ultimate goal is to go from bedside to bench and back so we can advance the care of our patients.
No, great, thank you. And this award is given to someone in the prime of their career, as I mentioned before. And so I'm wondering, do you have any people that you would consider part of your ideal mentoring team and or inspirations? And do you have some words of wisdom for the up-and-coming investigators?
I happen to be a late bloomer in terms of my own training. I have my roots in transcription factor biology several decades ago, and the mentors that believed in me at the time and took me under the wing were Dr. Ed Haber and Arthur Lee, who have passed away, and I was fortunate to have other mentors that fill in the gap as my transition to fellow to faculty a couple decades ago. Dr. Mukesh Jain, who had roots in transcription factor biology, and Dr. Peter Libby, of course, who's world-renowned expert in inflammation in the blood vessel wall and atherosclerosis. So I think the take-home message for trainees is really to identify a network of mentors that can help you guide as you transition in your career. And still to this day, I always bounce ideas off Dr. Peter Libby, who's been an inspiration for many of us in terms of what can be accomplished going from basic to clinical research.
Well, Dr. Feinberg, congratulations again.
Thank you so much. Very humble. Thanks for having me.
Absolutely.